Good morning. My name is Ellen Strong, and I've been here since 2004 as a curator and research zoologist in the Department of Invertebrate Zoology. What I want to talk to you about today is my work on cerithids, and to sort of give you a window on some of the challenges that are faced by malacologists describing and documenting the diversity of molluscan species. Cerithids are a moderately speciose family with about 185 species currently recognized as valid. They occur in tropical to warm temperate regions around the world. And this is primarily shallow water radiation. They occur anywhere from intertidal to subtidal depths down to about 80 meters, with some species that occur down to 1,200 meters. These are typically rather large, solid species, so ranging anywhere from about 1 centimeter to 15 centimeters in size. This is an example of one species showing the range of variation that you see in one species typically. And because of the fact that they are very solid, the fact that they are abundant and gregarious in shallow waters, easily accessible, they are usually well represented in museum collections. One of my predecessors here at the Smithsonian, Richard Haubrich, was also a scholar of cerithids, and he completed some of the last major revisions of this group in the 80s and 90s. And during the time that he was doing his work, the reigning paradigm at the time was of widely distributed, highly variable species. And this is one example showing Cerithium columna with many different shell variations distributed from essentially the Red Sea to Hawaii and places beyond. When I came to the Smithsonian, I didn't necessarily intend to follow in Joe's footsteps, Richard Haubrich's footsteps, but I've been able to use molecular data now to try to test some of these hypotheses about widely distributed species and variation in species in ways that weren't possible when Haubrich did his work. So what I'm going to talk to you about today is how this data is either rejecting or confirming some of our ideas about the distribution and species diversity in this group. The data that I'll be showing you today comes from across the Indo-Pacific. The circles shown in red are localities where I've processed specimens myself. In yellow are some additional sources of data, but the vast majority is stuff that I've collected myself. And gray is forthcoming sequences from recently completed fieldwork. And at this point, I need to acknowledge that I've been able to benefit from some of the expeditions that have been organized by Philippe Boucher at the Paris Museum. I've compiled to date a data set for about 600 individuals of cerithids. This is my tree. It's not important right now that you see details or names or structure or anything like that. I'll be zooming in to show you more details on the following slides for individual species. This is a data set for partial mitochondrial sequences of CO1 and 16S sequences. But I just wanted to give you the sort of the scale of the sampling that I have at this point. So here we have a species, Cerithium rostratum. This is a species that occurs across the Indo-Pacific from Madagascar to Hawaii. You can see some of the shell variation that's evident in this species, and these are representative shells that I've sequenced from across its range. And this is an example where the molecular data is confirming some of these ideas about broadly distributed, highly variable species. So in the upper left-hand corner, we have a branch of my tree, and all those little twigs are the individuals that I've sequenced. And the fact that those twigs are very short, very small, is indicative of the fact that there's very little genetic difference between individuals that have been sequenced from sometimes very vast distances across the Indo-Pacific. And this molecular result is consistent with the interpretation that Cerithium rostratum does in fact represent one highly distributed, highly variable species. We can compare that to a species that we find at depth. So Cerithium rostratum is an intertidal species. This is a species that occurs at great depths, down to 1,200 meters. And again, we find that the molecular tree is consistent with the interpretation that this is one highly variable, widely distributed species across the Indo-Pacific. But those examples are turning out to be the exception rather than the rule. Here we have an example where the molecular data is supporting 3 lineages, one in Hawaii, one in the Western Pacific, and one widely distributed species in the Southern Pacific. This is one of the more complex examples where we have a result that's supported by the molecular data showing 4 molecular lineages, one of which is actually more closely related to a species called Cerithium citrinum than it is to the other species currently referred to in Cerithium columna. So what do we call these species? I have molecular data for about 70 species from the Indo-Pacific with 40 species that have been sampled from more than one locality, and only 22 of those comprise a single molecular lineage. The remaining 18 species comprise 46 deeply divergent molecular lineages. And I am running out of time. I'm going to skip ahead here. But the point that I wanted to make here was that you can see that with these very complex patterns of many molecular lineages and overlapping geographic distributions, how important it is to have the sequence specimen. The hologenophore is a term that has been used to describe the sequence specimen. And in order to put names on these, we can then compare the hologenophores to the types. And this is the type of Cerithium columna. And with the type in hand, we can put the name Cerithium columna on this widely distributed species from the Southern Pacific. The other taxa we could refer to as dark taxa. These are things that don't have names, that are not currently referable to a currently identified species. But the other ones, what are they? Are they new? Do they have names? What are they? And the problem with snails is that there is a vast lexicon of names that occur in the literature. And we estimate that there are 300 to 500,000 names that exist in the literature, representing only about 82,000 that are currently considered valid, meaning that there's about 150,000 to 200,000 names that have been relegated as synonyms. And the term digging in the graveyard of synonymy has been used to describe the process of sifting through these names to try to apply them to lineages that we are recovering with our molecular trees. And this is a great burden and a great challenge for documenting and describing the diversity of mollusks. And I'm sorry I went long. Thank you.
We'll still let you have a question.
Thanks.
Why do different members of the same species make shells that look different?
No, that's a great question. Why is there variation in species? Is that the?
Why does different members of one species make shells that look different? Why aren't all the members of the same species shells that all look the same?
Right. Well, that's a great question, and we don't really understand the mechanisms of the maintenance and origin of diversity within species, and you don't find the same patterns across the mollusca. So there are some species that I'm working on that have large amounts of variation across their range, and other species that are highly constrained in their morphology. And we don't have a good explanation for why some species show this great constraint in the morphology, why some species show this great variation, and what is maintaining diversity in some and not in others. We don't know. Thanks.